Well, thank you very much, Paresma, Nikolett, and Shirley, for really outstanding presentations on topics that are fairly impenetrable to clinicians like myself. We have a lot of questions from the audience, so let me get to the first here. Let's see. The first one would be for Paresma. This is a CMC question. If a new formulation is introduced in the clinical trial, what is the expectation in terms of differences between the submission timings of the revised protocol and the CMC amendment? Any thoughts on that, Paresma?
Thank you for that question. I want to start off by just acknowledging that oftentimes we do see changes in the drug product formulation quickly during development. And what we often see is a variety of strategies with submission of that new formulation to support a revised or a new clinical protocol. What we would recommend is that the CMC information supporting a new or a revised protocol, if it's different from the original submission, that information be submitted either before submission of the clinical protocol or along at the same time as the new or revised clinical protocol. One thing that I would not recommend that becomes challenging from our perspective is if a protocol is submitted that proposes use of a new formulation, but the corresponding CMC information has not been submitted to the IND. So this oftentimes leads to us having to request that information from an FDA perspective from the sponsor. So that's kind of just information about the submission timing and strategy. I am also going to ask my colleague Nikolett Biel, who represents Pharmacology, Toxicology, to also provide some additional input from her perspective as well. So thank you.
Thank you, Paresma. So one thing to note from a non-clinical perspective is that typically when a new formulation is introduced in a clinical trial, sponsors often submit a single dose PK study in one species of animals just to demonstrate that the PK characteristics of both formulations are comparable, so there wouldn't be any exposure differences with the new formulation before administering it to patients. Thank you.
Thanks very much. Thanks, Nikolett and Paresma, for discussing new formulations. Nikolett, I have another one for you. The question is, the purpose of the investigator brochure is to adequately inform investigators participating in trials of a drug. And the individual is asking, what is the FDA looking for when they review the non-clinical portion of the investigator brochure during the 30-day initial IND review and for subsequent amendments to the investigator brochure? Any thoughts about what we look at? And
yes, thank you for that question. That's a very good question. So the investigator's brochure will summarize all the data that the sponsor submits to the IND. We review the original study reports. So, with a 30-day safety review, we're looking at proof of concept studies, whether the sponsor has demonstrated that the drug's mechanism of action is what they claim to be, that there is activity, from an oncology perspective, that they've showed activity, anti-tumor activity, whether in vitro, in vivo, or both. And then, of course, we assess the safety, through safety pharmacology studies and toxicology studies, and whether those studies are adequate to support a clinical start dose. I've touched on this during my presentation, but sometimes when 3-month toxicology studies come in, there might be a new toxicology finding that that may not have shown up in the 1-month study, in which case, this would be included in the investigator's brochure amendment. And we would look for that information and make sure that it's sufficiently addressed. Thanks again. That was a good question.
Thank you. So I have a question for Shirley. Food can be highly diverse based on its composition and may contain various proportions of carbohydrates, fats, and proteins. How do you account for this in terms of an ADME study? Any thoughts there?
Yes, thank you for that question, Leonard. So we have a food effect guidance which directs the developers of drugs to conduct food effect studies in the worst case scenario, which for most drug products is a high fat meal. So it is the best idea is to conduct a food effects study with a meal that contains very high fat content, and we do specify the different compositions of food that can be used. And for drugs where there may be a concern of exposure with food with low fat contents, you can conduct a study where you compare high fat meals to low fat meal on the effect of the drug exposure. So it just depends on the drug product itself, but yes, definitely the composition of the food or meal can have an effect on a drug product, and that is oftentimes formulation specific. So if a different formulation is developed, we recommend that a new food effect study be conducted with the new formulation. Thank you for that question.
Great. Another question for Paresma. The question is, what is your recommendation on the approach that industry should use to determine and ensure that batch data submitted are sufficient to support regulatory review and approval decisions? So this is the question on batch data.
Thank you for that question, Leonard. So at the time of an initial IND submission, our focus from the CMC perspective is really on safety, and we really focus on the differences between the submitted toxicology and clinical batches, and understanding the differences in kind of in the simplest example of of a small molecule drug substance and drug product, really focusing on the differences on the impurity profiles there. And so what we ask is that sponsors really outline the differences between the toxicology batches and the proposed clinical studies, clinical batches, and then provide any justification for why impurities may be greater in the clinical batches compared to the toxicology batches, and information on the levels that have been qualified of those impurities as well. So I think the other piece of this I want to clarify is that we have sponsors take very different strategies associated with submitting batch data. What we really want to see is assuring that the drug substance and the drug product can both be manufactured. And this doesn't necessarily mean that the clinical batches have to be manufactured and submitted. Ideally, that would be the case, but even submission of engineering batches of the drug product may be sufficient to just demonstrate that the product can be manufactured. And we do oftentimes see times where a sponsor may submit both that the toxicology in the clinical batch will just be the same. So they may only submit one batch. So it really depends on the study that's being proposed. I hope that answers the question. Thank you.
Thanks very much. I think that's a really insightful answer. Let's turn to Nikolett again. This one's slightly shift in focus. The question's about an immunotherapy toxicology study. The question is whether that's dose independent. How is the clinical start dose decided on with a fixed dosage? Perhaps you can unravel that for us.
Yes, thank you for that question, Leonard. Again, good questions. So hopefully I understand the question properly. So for immune modulating agents, the start dose is typically determined based on the minimal anticipated biological effect level, or MABEL, which really takes into account all the available data that the sponsor submits, including pharmacology studies, especially that are relevant to the mechanism of action of the drug and potential for cytokine release syndrome, and toxicology studies that are submitted, and any clinical experience that may be available with a similar drug, we can take into account. Sponsors often use PK modeling, taking into account, all the data that I just mentioned when they propose a start dose. And as far as dose escalation with immune modulating agents or biological products in general, following a half log or less increments is generally acceptable. So that's the short answer. I hopefully answered the question. But a lot goes into it. It depends on each product, but really it takes into account all the available non-clinical data to determine a start dose. Thank you again.
We are apologies. We are having some difficulties. Leonard, we had put you on mute because we were getting background noise. Could you go ahead and check to unmute yourself before you proceed? Thank you.
Thanks, Jeff. It looks like we were having a little bit of trouble with audio from Shirley, so I'm looking for another question here for one of the other folks. Nikolett, there's a question which I'm not sure if there's enough information to be able to answer, but the question is, what if the reprotox is not available, if the reproductive toxicology is not available? Any thoughts on that?
Yes, thanks, Leonard, for that question. So often, at the first in human IND stage, reproductive toxicology studies are not available, and that is fine. The potential for embryo-fetal toxicity may be expected based on the mechanism of action of a drug. For example, drugs targeting rapidly dividing cells, we would expect there to be a potential for embryo-fetal toxicity. Often clinical trials will include recommendations for contraception to prevent pregnancy during and for a particular time after the last dose of a drug. And then as drug development proceeds, before a marketing application would come in, the reproductive toxicology studies would be available. And we can use that information to recommend contraception timings for, when the drug is approved to include in the drug label. Thank you.
Thanks, that makes excellent sense. I'm wondering if we've got Shirley back. Shirley, if you're available, again, the question was on in what scenarios would conducting drug interaction studies be appropriate?
Hi. Yes, I thank you for that question, Leonard. I am back. For drug-drug interaction studies, we do often recommend an in vitro study approach first. to determine if a drug might be a substrate for or a modifier of certain drug metabolizing enzymes. So we recommend in vitro screening strategy prior to decisions on whether or not to study the drug in a clinical setting. So if it comes up as a positive in the in vitro screening, we recommend clinical trial investigations and drug interaction studies, as they're called, to further evaluate the magnitude of the effect in in vivo setting. Thank you, Leonard.
Great, thanks for a good answer there. Paresma, one for you. What kinds of CMC issues are noted by FDA during the review of a first-in-human phase 1 IND study? Any thoughts on that?
Thanks for that question, Leonard. I want to say that I very briefly covered this in the presentation, but I'm happy to discuss some of the CMC issues that are often considered safety concerns that may lead to holds or clinical holds. I do want to note by just kind of clarifying that for a phase 1 first-in-human IND submission, the review team is really focused on safety issues that may lead to concerns for CMC of identity, purity, strength, or quality of the investigational drugs. And that's based on our regulatory requirements. So some of the common or some of the CMC issues that we do see includes not having sufficient characterization of the drug. And in particular, when thinking about drugs that are manufactured using fermentation or recombinant technology, this includes not having sufficient characterization data for the cell banks for manufacturing. One of the other common areas is lack of information on the impurity profiles, and I mentioned this previously, but that comparison between the toxicology and the clinical batches. So if there are impurities in the clinical batches that are not supported by or qualified by the toxicology studies, that may lead to some concerns. The other ones are oftentimes related to the drug product formulations, where the formulations have not been optimized and may lead to things like precipitation of the drug. And this oftentimes leads to concerns that we don't know the strength that's actually being dosed, and the obvious safety concerns associated with the formation of particulate matter in the drug product. And this may be in the drug product itself, if it's in the solution, or when being administered as part of the in-use compatibility studies that are submitted for drug products. So those are just a few of the examples that we do kind of see and want to assure sponsors are looking out for. The other thing I do want to note is it's really important, and we can't emphasize this enough, that sponsors consider meeting with the agency with a pre-IND meeting, particularly for the first-in-human submissions, because I think it really helps to assure that their clinical protocol is also on target, and that the dose strengths that they're going to need for the clinical study have been thought about, as well as any potential hold issues can be identified from both CMC, but also other disciplines as well. Thank you.
Excellent. Thanks for a great answer there. Very important stuff to consider. Shirley, one for you. When during drug development would you recommend a food effects study be performed, and why? Thoughts on that?
Great. Thank you for that question, Leonard. So we often recommend food effects studies be completed as soon as feasible. And that is because we need to know whether food will alter the systemic concentration of a drug. And we often need this information prior to additional clinical trials that come down in, phase 2, phase 3, those further downstream clinical trials. It is important to know what food might do to the bioavailability of a drug so that we can help decide what to instruct patients and how they take their medications. So the sooner we have that information, the better, in order to de-risk drug development later on. Thank you.
Thanks very much, Shirley. Here's one for you, Paresma. At what stage during clinical research should the drug product manufacturer be identified?
Thanks for that question, and this is a really good question. The information about the drug product manufacturer, the name and address, is actually a regulatory requirement for IND submissions from the time of opening that IND. And so this is actually part of our CFR regulations. The addition, deletion, or change of any of that manufacturer information after that initial phase 1 or original IND submission should be reported to the agency. What I want to point out here, and maybe where this question is going, is, when do you have to have that final commercial drug product manufacturer identified? And that is kind of a different question. So there's a regulatory requirement that whoever is making that drug product be submitted in the IND, and as changes happen to that manufacturer, that information also be submitted to the IND. However, the final drug product manufacturer that you may be planning to use for a commercial or a marketing application may not be finalized until later in development. Oftentimes, this is happening at around the end of phase 2, and sometimes even around the time of the pivotal clinical studies being run or prior to marketing. So what I would say is, as those later stage changes to drug product and or drug substance manufacturing sites are happening, that sponsors come and meet with the FDA as part of maybe a CMC focused type C meeting to discuss their proposed manufacturing plans as they get closer to marketing. Thank you.
Thanks very much. Thanks, Paresma. Nikolett, another question for you. This is sort of a different different emphasis. What is the rationale for selecting higher order mammals, for example, canine or primate, for IND enabling safety studies?
Thank you for that question, Leonard. Yes, so we typically expect a rodent, 2 species, a rodent and non-rodent toxicology studies to support an IND. The reason being that sometimes higher order mammals, dogs or monkeys, may be more sensitive to certain drugs. It could be a different way that they're metabolizing the drug. And so this could lead to a lower clinical start dose that may be supported by a non-rodent species compared to the rodent species. Also, for biological pharmaceuticals, many times the rodent is not a pharmacologically relevant species, and maybe only the non-human primate may be the only pharmacologically relevant species. And so that would be the species to conduct a toxicology study to support, a clinical trial and assess a safe starting dose in humans. Thank you.
Great. Excellent question, and certainly a very comprehensive answer. I've got a question which is addressed both to Shirley and Nikolett. Does the FDA require all the laboratories where these preclinical pharmacology toxicology studies are performed to be good laboratory practice certified, GLP certified? Maybe start with Shirley. What's your thought on that? And then maybe we can move to Nikolett.
Yes, thank you for the question, Leonard. Actually, this question is much more suited for Nikolett, so I'll let her go ahead and take it. Thank you.
Sure. Thank you, Shirley, and thank you for the question. Good question. So typically pharmacology studies, using cells or mice, for example, tumor-bearing mice, they're not expected to be GLP compliant. However, toxicology studies, safety pharmacology studies, genetic toxicology studies, reproductive toxicology studies should be GLP compliant. Oftentimes safety pharmacology studies can be included as part of the toxicology, pivotal toxicology study, or some sponsors may decide to do separate standalone studies, but they can be rolled into general toxicology studies as well. Thank you.
Thanks very much. I think we're almost out of time. Let's get one more question in. This one is for Paresma. Let's see. It is understood that drug product batches manufactured for clinical trials are typically still going through the improvement process that require adjusting specifications. Should the level of good manufacturing practice for clinical batches be the same with commercial batches? If not, please advise where GMP focus for clinical batches should be. Any thoughts on that, Paresma?
Hi, Leonard. Thank you. That's a really great question. And I will start off by just saying, yes, it is very often that we see significant changes to chemistry, manufacturing, and controls from the original submission of an IND as you move through clinical development. So it doesn't always happen, but it is a very common aspect that is changing during development as the formulation is kind of being optimized. What the question is really getting to here is about GMP expectations here for through phases of development. So what I want to highlight is the regulations for cGMP are actually outlined in 21 CFR 210 and 211. Those regulations are really focused on commercial manufacturing of products and may not be appropriate for phase 1 clinical studies necessarily. But for phase 1 clinical studies, the FDA has actually published a guidance, and I highlighted that guidance on my resources slide. That guidance is specifically titled cGMPs for phase 1 investigational drugs. And those focus areas for INDs in phase 1 are in that guidance, and the requirements include thinking about well-defined and written procedures, adequately controlled equipment and manufacturing environments, and accurately and consistently recorded data from the manufacturer. So what I would say is phase 1 is where the expectations can be slightly different and outlined in the guidance. For phase 2 and 3 manufacturing, those phases of development are subject to the CFR parts 210 and 211 and the regulations for commercial manufacturing that are outlined in those parts of the CFR. So thank you.
Great. Thanks for an excellent and comprehensive answer. So I guess that brings our session to a close. Again, thanks to our speakers for really an outstanding review of the non-clinical and early clinical studies that we may come across, and for a really excellent review response to the questions. We will now take a half-hour break, and we will return half an hour's time. Millie Dougal will be leading that session. Thank you very much.